AS Level Biology 9700, Paper 2, Variant 1, October, November 2025. And in this video, we are going to discuss question number 4. And question number 4 is Figure 4.1 is a photomicrograph of a copepod. These animals are found living in seawater and in freshwater environments. You can see the copepod in the picture. So the outer surface of a copepod is covered in a layer of the polysaccharide chitin. Chitin is outer protective covering. So figure 4.2 shows part of a chitin molecule. Chitin is a polysaccharide. Polysaccharide means type of carbohydrate. So 4.2 shows part of the chitin molecule. You can see draw a circle around a polysaccharide bond in figure 4.2. And you know that in class we had studied about the formation of the glycosidic bond. Glycosidic bonds are formed as a result of condensation reaction in which on the formation of one glycosidic bond, one water molecule is removed. So here you can see the glycosidic bond is formed between two monomers, between two molecules of chitin molecule. You can see over there. So this is the glycosidic bond. So we have drawn the circle. Now the next part of this question is in aquatic environments, Vibrio cholera can live on the surface of copepod. V. cholera secretes enzymes to hydrolyze within to its N-acetyl glucosamine monomers. These can be broken down to provide carbon, nitrogen, and a source of energy. Draw the monomers that is found when chitin is hydrolyzed by V. cholera. So this is the type of bacteria which cause cholera. So I am going to move towards the that question because we have to uh, draw the monomers right now. So in this diagram, in this figure 4.2, they have shown two monomers. So we have to draw these monomers. We have to hydrolyze them. So when they get hydrolyzed, they are produced as a result of condensation reaction that the one water molecule is removed. When water is added into them, these molecules are hydrolyzed. So when they get hydrolyzed, this bond will be broken down. The glycosidic bond will be broken down. All they are the glycosidic bonds. So they have been shown only two monomers, but when they will break down, they will also be uh, removed from here. So I'm going to draw these on this given space. So moving towards the given space where we have to draw these monomers. So instead of O glycosidic bond, there will be the OH molecule over there. So first of all, when we are going to draw, we have to draw the first the OH, same as it has been shown in the diagram 4.1. CH2OH, and then here you can see NH, C, NH, CH, and CH3. So this is the one monomer. So we can draw both of these monomer in the same way. So it has been, instead of glycosidic bond, there is OH molecule over here, because the glycosidic bond has been broken down by the uh, addition of water molecule. The other monomer which was shown in the picture, that was in this way. Again, it will be addition of OH molecule instead of only oxygen molecule. OH image, same. They will be same, and they they will be opposite to each other. Their direction will be opposite. So this is one monomer and this is the other monomer. They were joined together with a glycosidic bond in that figure 4.1. Now, moving towards the next question. And next question is, V. cholera is a pathogen that causes cholera. Scientists studied the transmission of V. cholera in groups of people living in an area where there is a high number of cases of cholera. Some families living in this area filtered their water through several layers of folded fabric from old clothing. The folded fabric trapped particles and organisms larger than 20 micrometer. 20 micrometer, very small in size. So the scientists recorded the number of cases of cholera in families that filtered their water through folded fabric and compared this to the number of cases of cholera recorded in the families which did not filter their water through the folded fabric. So it was very, you can say, the method with the help of which they recorded, with the help of which they filtered the water. And now we can observe this graph. This is a bar graph on which on x-axis they have given the water not filtered and water filtered through folded fabric. So number of cases cholera per 1,000 population. You can see the number of cholera cases are high in case when the water was not filtered by the people. On the other hand, you can see this graph, small bar, in which the number of cases of the cholera per 1,000 population are very uh, about half of the cases with cholera. So they were treated with they treated the water, they filtered the water. So our question is, suggest possible explanations for the results shown in figure 4.3. So figure 4.3 suggest possible explanations. So we are going to write down the explanation for this. So they are, uh, first of all, I will explain and then I will write the answer. Here you can see that the filtration method is very effective. So this question shows the effectiveness of the filtration, and the graph shows a significant reduction in cholera cases. So physical by the this by making use of this folded fabric, it acts as a physical barrier. While individuals Vibrio cholera bacteria are usually smaller than 20 micro units, so they often attach to larger organisms. So cholera bacteria frequently cling to copepods or planktons. They are larger. So by trapping these larger organisms, the fabric effectively removes the vehicles carrying the pathogens. So the graph shows that cases were halved but not eliminated. They were halved. Already discussed that they are almost half. So it means some free living Vibrio cholera bacteria are small enough to pass directly through the fabric layers. So the fabric might become saturated with pathogens over time, or users might accidentally touch the clean side of the cloth with contaminated hand. It means that they are totally not removed, but after some time they get linked with the people who are filtering them. So cholera can also spread through contaminated food or person to person contact, which water filtration alone cannot prevent. So now I can write all this, which I have explained to you. So I can write this. The folded fabric acts as a physical barrier Vibrio, cholera, diphtheria are usually very small, about 20 micron. So cholera frequently diphtheria frequently cling to copepods or other planktons, water planktons, that are larger. By trapping these larger organisms, the fabric effectively removes the pathogen. So, some free swimming can also pass through the fabric or fabric become concentrated with them, which can cause cholera. So cholera can spread through contaminated food or person to person contact. So these were the possible suggestions and explanations for this question. Which water filtration alone cannot prevent? And you can see half of the bacteria were removed by the filtration method. So it was about this question. Now moving towards the part C of this question. The World Health Organization recommended the use of an oral cholera vaccine to protect people living in an area where a outbreak has occurred. So people who receive an OCV and make changes in their behavior are less likely to have a serious case of cholera. Describe one change in behavior that a person can make other than purifying water to help prevent serious case of cholera. So here behavior change uh, to remove the cholera or to protect ourselves from the cholera. The behavior change can be improve. You can say the possible answer. We have to write the possible answer. That is the washing hands with soap specifically after using the toilet or before handling and preparing food. So ensuring food is cooked thoroughly and eaten while not, or washing raw fruits and vegetables with treated water. So in this way, we have to write only one possible answer. So we can write washing hands washing hands with soap after using toilet. Or another can also be wash wash the fruits and vegetables before cooking. So okay. Now next question is, the antibiotic tetracycline is used to treat cholera. However, some bacteria that cause cholera have evolved resistance to this antibiotic. Scientists have reported that resistant bacteria have an extra protein in their cell surface membrane. This protein has been found to use ATP. ATP, the energy which is produced by the body by the mitochondria, suggests how the presence of this protein in the cell surface membrane gives weak cholera resistance to tetracycline. So we have, first of all, we must know what is the mechanism of antibiotic resistance. So this is you can say we have, again, this question is, in this question we have to suggest. So we can see that uh, we need to apply our knowledge of cell membrane and transport to a new scenario. So according to this question, we can say the protein likely act as an active transport pump. So the protein, which is the extra protein, it acts as a active transport pump, so which uses ATP. So we should write if the drug cannot stay inside the cell, it can also do its job. So I am going to write this question uh, with detail because only 2 marks, but you can do it according to your writings. So the protein uses acts as an active transport pump. The protein uses energy from ATP, hydrolysis, to actively pump the tetracycline molecules out of the bacterial cell against a concentration gradient. This keeps the internal concentration of the antibiotic low, allowing the cell to survive even in the presence of the drug. So because it pump out the tetracycline out the cell. So next question. For some vaccines, there may not be an effective secondary immune response when a person is infected by the specific pathogen. The antibodies that are produced do not act on the pathogen. This is known as immune evasion. Evolution of resistance of bacteria to antibiotics occur more frequently than immune evasion. Suggest why bacteria evolve resistance to antibiotics more frequently than vaccines. Resistance to antibiotics more frequently than vaccines that lose their effectiveness in protecting against bacterial pathogens. Again, the question is to suggest. So we have to suggest that why it happens. So we know that the vaccine typically, uh, when it when it is used against the different pathogens, so what happens with it? These vaccines trigger the production of many different types of antibodies that recognize various parts of a single pathogen. For a bacterium to evade a vaccine, it would need to mutate, multiply different surface proteins simultaneously. So, in contrast, an antibiotic usually targets one specific metabolic pathway or enzyme. A single mutation and that one target can render the drug useless. So antibiotics are, the passive chemical tools. If the bacteria change the shape of the target position, so what happens? Uh, wait, okay. So the immune response protein, the antibiotic can no longer bind. So the immune response is active. The body produces a diverse army of beta cells and T cells, so making it much harder for a pathogen to hide from all of them at once. So we are going to write this question in this way as, because it is also of 2 marks, I have given too much detail, but we have to write a little brief. Usually have only one specific target site. Site of action in bacteria. So a single mutation can provide resistance. Whereas vaccines cause the production of many different antibodies which target multiple different antigens, making it much harder, making it much harder for the bacteria to mutate on targets at once. So it was all about question number 4. So at the end of this, I would like to say that please like, share, and subscribe my channel. Thanks for watching.